All right, so we will get the review started. Hope everybody had a good weekend. So the test is going to be for us on Wednesday, 8 o'clock. It's 100, I'm sorry?
It wasn't 9?
No, at 8 o'clock,
Okay.
8 o'clock, yep. You got 2 hours to take it. 8 to 10 is when we have the room, just like today. So it's 100 questions. Hopefully you got the emails I sent out to you to give you ideas of what you're shooting for. It is not super hard. It's just a lot of material. So we're going to go over, starting at blood, chapter 14. We'll come back to 13, which is the endocrine system, when we get to the urinary system. But just to give you, again, some foundational stuff that you got to bring with you from 210. So this is the last test in this 2-semester series, 210 and 211. So in 210, you started off with the building blocks, atoms. Subatomic particles make atoms, atoms make molecules, molecules macromolecules, macromolecules organelles, organelles cells, cells tissues, tissues organs, organs organ systems, and then the organ systems make you. And as you've gone through 210 and 211, that has been kind of the idea of looking at how they're built up and then get to the organ systems. After that, you got into just directional terms and how to communicate with other providers. You still need to know that for sure. Then you went into chemistry, which everybody's favorite chapter was chemistry, but now looking back, you should be able to realize how important that was. Things like pH, you're never going to get away from. You need to understand about pH. pH is a huge issue with our bodies. Even the most healthy people have to deal with regulating pH. So, and even before we got to that, in that first chapter, you talked about homeostasis. Pretty much every system you've looked at since then is talking about how it affects homeostasis. Homeostasis is how our body keeps everything in a normal functioning position. And so pH is, we have the normal negative feedback mechanisms, which we call buffers, to help keep us in a normal pH. Remember, pH is 0 to 14, 7 is in the middle. Middle means it's perfect water, as many hydrogens as you have hydroxyls. From 0 to 7 is hydrogens are excess, 7 to 14 are hydroxyls. 0 to 7 is acidic, 7 to 14 is basic, and in our body it's called alkaline. Now, our body is slightly alkaline. 7.4 is called physiological pH. We are supposed to be in this little 1-point range with 7.4 in the middle. If I get too low, I go through acidosis. If it is because my lungs aren't functioning, it is respiratory acidosis. If it's because my kidneys aren't functioning, it is metabolic acidosis. If my pH starts getting too high, I go into alkalosis. If it's because of my lungs, it's respiratory alkalosis. If it's because of my kidneys, it's metabolic alkalosis. So again, all of these things kind of get brought up, and that's why I keep kind of trying to remind you, you've got to remember stuff as you keep going forward. So the next chapter, we looked at cells, and we kind of talked about how cells were put together. And actually, going back before that, the last part of that chapter to the chemistry, you started talking about how molecules are formed, and we talked 2 different ways, ionic versus covalent. Ionic is where there's an absolute loss of an electron, causing the little atom to become charged. They're called ions in that chapter, but as you went forward, they started being called electrolytes, and occasionally they're called minerals. Sodium is the major one, right? But there's all kinds of them, calcium and potassium and whatever. Those are ions. They are minerals, and we call them electrolytes. They're very important in how the body works because they are the thing, like for action potential, still going back to 210, that's what the ions do, is they cause the electrical charge to go down the axon. Then you got into about these big molecules called organic molecules. Again, 211, you got to know 4 organic compounds. You've got carbohydrates. Its building blocks are called monosaccharides. The main monosaccharide is glucose. You have proteins. Proteins are the things that make all of the enzymes and the antibodies we looked at. The building blocks are called amino acids. The building of a protein goes from amino acids to peptides until finally we have a polypeptide and then a protein. Then you have lipids. The main dietary lipid is a triglyceride. Lipids are considered fats. They are the thing that don't function well inside a watery environment. All lipids have to have some sort of protective mechanism to move around in our body. Right? Again, we looked at that this semester. We called most of them lipoproteins, but there's all kinds of little things that have to happen for lipids to be broken down and move around in our body. And then finally, nucleic acids are the building blocks of things like DNA and RNA. It's nucleotides that build those up. All right, so as you started moving forward and looking at the different organ systems, you see how they fit together. And in this class, it is much more about the physiology of stuff. Skeletal system, muscular system, and even the nervous system, you're looking at basic areas and still basically anatomy. So moving into this semester, we started with blood, and you'll see, and hopefully have seen, that every chapter since that first one deals basically with blood. Whether it's the respiratory system charging blood, whether it is the urinary system cleaning blood, whether it is the digestive system loading blood. It is all about blood as the main mechanism to get things around and keep our body stable. And so when we were looking at this, so chapter 14, we started out with blood, and we talked about how blood is made up of two components, and it's very important to know this. 45% are formed elements, 55% is plasma. Now going back to that chapter 2 and 210, there was a really important thing we learned called osmosis. So many things are dealing with osmosis. So osmosis is all about concentration of fluids next to each other with a semi-permeable barrier. So if I have something that is 55% plasma and 45% formed elements, like in the blood entering the glomerulus, and then as the plasma is separated, now it is down to, let's say it is 40% plasma versus 60% formed elements. This is concentrated, this is dilute. Once you set that up, you should at this point know there's going to be some sort of osmosis that goes on here. There's going to be a movement of water. You should start being able to see that at this point, right? Whether it is, again, now I was using the kidneys as an example, or it could be in the capillaries of the regular tissues as an example. But you got to start being able to kind of have that in the back of your mind so that when you're reading something or studying something, there should be little triggers in your brain going, boom, hey, I should look for osmosis here. I should look for a movement of water here. I should know fat-soluble things can move through cell membranes versus water-soluble. All of those little base things you need to carry with you because it's going to make it so much easier, or said another way, it's going to be so much more difficult if you don't. If you keep thinking you have to relearn this over and over and over again, it's going to get to a point where you won't be able to keep up with it. You've got to have the material and move through it. Does that make sense? All right, so the concentration of blood. Now, formed elements, we've got three formed elements: platelets, red blood cells, white blood cells. Red blood cells are by far the most common of the formed elements. Their job is to carry oxygen. White blood cells, there are 5 types of white blood cells, and by the way, you need to know the scientific name for these. Platelets are called thrombocytes, bless you. Red blood cells are called erythrocytes. White blood cells are called leukocytes. Of the white blood cells or leukocytes, there are 5 types, and these are divided up into ones that do not have apparent granules called agranulocytes, and then the ones that do have apparent granules called granulocytes. The neutrophil, the eosinophil, and the basophil are the granulocytes. The only one I want you to know anything about the granules on is basophil. You should know it is histamine and heparin, and at this point, you should know histamine is an inflammatory chemical. It's going to cause vasodilation. Heparin is an anticoagulant, stopping blood from clotting. And so the basophil is the least common. Again, we said basal means bottom. And as you're going through, anytime you hear basally located or basal something, you should automatically think it's the bottom of something. Eosinophils have red granules. Neutrophils are the one of the two phagocytic cells in our white blood cell system. It is by far the most common of the leukocytes. Then we get into the agranulocytes. Monocyte is the largest. It also has another name if it leaves circulation, and it is called a macrophage. It is the second of the phagocytic cells in our body. And it is designed to come in and clean house. Neutrophils are the most common. They come in like little gnats and bother everything, and they start cleaning up. But the monocytes or macrophages, when they come in, they really take care of business. And then finally, these called lymphocytes. The lymphocytes are the T and B cells of our body. These are what we would classically call our immune system. All right? They're the ones that are going into and protecting us, and we'll look at those in a little bit. On the plasma, it is important to remember we have several different things. Water is the most common of the substances in plasma, but a very important one are the plasma proteins. We have one called albumin, which is designed to set osmosis. It is a big molecule that doesn't leave the circulatory system when blood goes through a capillary. And so it causes a very big osmotic pull on the interstitial fluid to be brought back in. Globulins, especially the gamma globulins, are what we look at in a minute, called antibodies. And then finally, fibrinogen. And again, when you see OGEN in something, that means it's a precursor to whatever's in front of it. So fibrinogen is an inactive form of fibrin. Fibrin is designed to clot. Now, the other things, nutrients, vitamins, hormones, and gases, we've talked about. The wastes are the things that we are getting rid of, that we talked about towards the end of the semester. So all of this kind of is stuff that we end up touching on through the semester. Now, when we go to the doctor, the doctor will do what's called a hematocrit, where they spin the blood, whole blood around, and divide it up into its two portions to look to see that there is a 55-45% split that is healthy. If it is below 45% for the formed elements, that usually is a sign of something called anemia. So the kidneys are really kind of controlling the creation of red blood cells. Hematopoiesis is just a catch-all name for creating any of the formed elements. If we're talking about red blood cells specifically, it's called erythropoiesis. And so erythropoiesis, to fast forward when we were looking at the urinary system, is started by a hormone released from the kidneys called erythropoietin, which causes erythropoiesis. They all come from a common stem cell. The stem cell in blood is in red bone marrow. All of them follow a pretty distinct path except for one type, and those are the lymphocytes. Lymphocytes follow the lymphoid pathway. Again, that's not going to be something I'm going to test you on, but I'm just pointing it out because lymphocytes are special. So in red blood cells, which are called erythrocytes, we have a molecule called hemoglobin. Hemoglobin is what holds on to the blood and oxygen and carries it. It does not have a nuclei or a mitochondria, and we went over why, and I hope that makes sense. It's not designed to replicate. We do not want red blood cells making their own copies. We want that controlled by the stem cells and in control of the urinary system, liver and spleen, monitoring these red blood cells. The other thing is the mitochondria uses oxygen, and since we're trying to bring the oxygen to the tissues, we don't want a mitochondria in there. And kind of go back to reviewing some things on 210, we did look at it in here. Cellular respiration, you definitely need to know cellular respiration. Cellular respiration happens in 3 steps and in 2 phases. The first step is the first phase. The first phase is called anaerobic because it doesn't need oxygen. It happens as soon as glucose enters the cell. It is called glycolysis. Glucose is split. If oxygen is present, we can then get into aerobic respiration, which happens in the mitochondria. There are 2 steps to that. The very first one is called the citric acid cycle. The second step is called the electron transport chain. All of that is designed to create this molecule called ATP, which is our energy, right? So that is why we carry oxygen, because we need it to unlock the energy that's in glucose. If you do not have oxygen, you will die quickly, right? And it is because we cannot survive simply on glycolysis. We have to have cellular respiration getting into the aerobic part. Now, if we have low oxygen carrying capability, we have a disease called anemia. Red blood cells are constantly being monitored, and they're only really going to live for about 120 days. After that point, they are no longer going to be able to carry oxygen efficiently, so we remove them. The liver and the spleen are the areas that remove them. So hemoglobin is made up of a couple of parts. Heme is the part that holds on to oxygen, and iron is the electrolyte mineral ion that is the key to hemoglobin that holds it all together. Right, so our body recycles that pretty quickly. Now, with this, I hope you understand most of these terms. Diapeptides means that the white blood cells are moving through the capillary wall to go to the tissues that are calling it. The thing that is calling it is called positive chemotaxis. Chemotaxis is the chemical message that the cells that are in trouble send out, and that's what's going to cause the white blood cells to squeeze through. Phagocytosis, again, is engulfing and digesting these pathogens. Again, neutrophils and monocytes, which are also called macrophages, are the 2 big ones. And so of those terms, again, I hope you just understand them. I wouldn't expect to see a whole lot on that. But I do expect you to understand them. You really need to anyway. Again, going through this, not worried too much about those. You're not going to be able to identify them, no pictures, but you do need to know a little bit about them. Now, when we do a hematocrit, after we look at the ratio between plasma to formed elements, we then will look at the formed elements, we put it on a slide, and we do what's called a differential. A differential is when I look at all of the white blood cells that I can, try to count them, and look at their ratios, see where they stand. And if we have things that are increased or decreased compared to what they are supposed to be, it gives us an idea of whether it's an allergy or a bacterial infection or something like that, a viral infection. So we can kind of tell. It doesn't tell us exactly what's going on, but gives us an idea. But that's called a differential. Now, the idea of blood, remember, blood is mainly water. 55% of whole blood is going to be blood plasma, and of that 55%, over 90% is water. Now, again, the little plasma proteins, gamma globulins, we'll talk about in just a minute. They are antibodies, and so they're a big deal. So, hemostasis is a term that means stoppage of bleeding. There's a 3-step process. Again, with this, I don't know that I could, if I've only got, 10 questions to pull from this chapter, going through the process of the stopping of bleeding would be a pretty hard thing to just figure out how to even ask about it. But you need to know that in the last step of this blood coagulation, there is a change from fibrinogen, which is a plasma protein, to fibrin. Okay? I hope that the idea of how this works makes sense. If I have a cut in a hose, the small muscle, the smooth muscle that's in front of it is going to constrict, causing it to decrease blood flow to that injured spot. In the injured spot, I'm going to have platelets start sticking to it to start closing that opening, again, limiting blood. And then the act of fixing that problem happens, and we call it coagulation. Right? It is a process. There is two types of processes that start, they'll have a common pathway, but we have extrinsic and intrinsic, outside and inside. The only thing I want you to know is hopefully you know that extrinsic means outside and intrinsic means inside. In order for a clot to be dissolved, we have to release something called plasmin. Fibrin, which is going to form this clot, is almost indestructible. The only thing that can get rid of it is this thing called plasmin. Now, if this goes wrong, you can have different problems. A thrombus is just an abnormal blood clot in the blood vessel. An embolus is when it breaks free and starts moving. Thrombosis is when this thrombus blocks a vessel, and that happens, we have what's called infarction, cell death. All this is saying that I quit receiving oxygen. That's how important oxygen is. If I block the oxygen off from an area of tissue for even a few seconds, it can cause major damage. Within a minute, you're talking about tissue death. Embolism is when this embolus, which is broken free, blocks a passageway, and so again, ends in infarction. And finally, atherosclerosis is the accumulation of fat inside the lining of the artery. Now again, we're hitting the highlights of stuff. I've got there's a lot of stuff in here. We can only look at the highlights again. So in blood typing, we're looking at red blood cells, and red blood cells will have markers on their surface called antigens. Now, we have antibodies for all kinds of different things, but we have specific ones for blood. And so we only have two true antigens. We have a third one that's added on, but it's A and B and RH. A and B is what I want you to concentrate on. RH, if you can kind of separate it, it's really simple, but if you try to learn them all together, it can get complicated. Blood cells are going to have A or B, or both AB or neither, but there's still only two antigens. There's 4 ways it can present, but only 2 antigens. Whatever antigen you do not have, you are going to have antibodies designed to look for it. And they will be called anti-whatever antigen it is. So we really only have 2 antibodies we're talking about with this. So I've got an A and B antigen. That means I have an anti-A and an anti-B antibody. Whatever antigen I don't have, I'm going to have an antibody that's designed to look for it. So when we start mixing them up, we have A, type A, which means I have an A antigen, which means I have an anti-B antibody. If I'm type B, I have B antigens, and that means I have anti-A antibodies. If I have both A and B, that means I am type AB. With that, I cannot have either one of the antigens. Now, if I am O, which means I don't have either A or B antigen, that means that I'm going to have both antibodies. Whatever antigen I don't have, I will have an antibody designed to attack it. And that, if you can remember that, it all falls into place. With the RH, it is simply I have it or I don't. Now, in the ABO system about A and B antigens and anti-A and anti-B antibodies, those are present at birth. There is no antibody that you are born with that would attack RH. So RH ends up being like a virus to you. you have to have two contaminations. Now, when we went into the lymphatic system, we talked about we have primary response and a secondary response to an invader. That's how our body treats the Rh antigen. If I have it, I can receive any type of blood. If I have it, I'm a positive blood type. And if I'm a positive blood type, as long as it matches on the ABO system, I can receive any blood. I can receive both positive and negative, as long as it matches the ABO system. If I am negative, I can only receive other negative blood types. Again, as long as it matches with the ABO system. But it takes two cross-contaminations to become a problem. So that's the idea of this. Now again, this is a review, so we're just kind of moving through it pretty quickly. So we go from looking at the blood to looking at the pump that's going to push it around and the blood vessels which will carry it. So this is kind of considered the cardiovascular system, the heart and the blood vessels. The heart's this little cone-shaped muscle with little compartments right in this area called the mediastinum. It sits a little farther over to the left. It is going to have a series of vessels that are either going away from it or coming to it. In this, again, remember, arteries always go away from the heart, veins always come to the heart. It doesn't matter if it's oxygenated or deoxygenated. It's all about the direction. All arteries go away from the heart, all veins go to the heart. So when we start looking at this, we have two circuits. We have a pulmonary circuit, which carries oxygen poor blood from the heart to the lungs. Those arteries are going to be blue on a model. When it goes to the lungs, it picks up oxygen, drops off carbon dioxide. We go through that period in the respiratory system where we talk about the bicarbonate comes in and goes through from taking that, combining it with hydrogen, making carbonic acid, and it breaking down to carbon dioxide and water, and we breathe it out. When that happens and it picks up oxygen, it goes back to the heart. And now the blood that's going back to the heart is oxygen rich. So those are called veins, but they would be red on a model. That's the pulmonary circuit. The one that we study in high school and stuff is basically the systemic circuit. This is going from my heart out to my body. In this, arteries are red, veins are blue, because I'm carrying the oxygen out to the tissues and then returning back to the heart. Now the heart has a couple of chambers. We have upper chambers called atrium, lower chambers called ventricles. The ventricles are the pump for the system. There are valves between the ventricles, between the atrium and the ventricles and the vessels they pump into. On the right side, we deal with deoxygenated blood. From the right atrium, it's going to go through the tricuspid valve to the right ventricle. When the right ventricle squeezes, it pushes the blood through the pulmonary, the semilunar valve into the pulmonary trunk. On the left side, we deal with oxygenated blood. So the left atrium, the blood's going to go through the bicuspid or mitral valve into the left ventricle. When the left ventricle squeezes, it's going to push that blood into the aorta. And to get there, it goes through the aortic semilunar valve. And so that's the flow of the blood to the heart. Now, the vena cavas are going to be bringing blood to the right side. The superior vena cavas bringing the deoxygenated blood from my arms and my head to the heart. Then everything below my heart is coming up on the inferior vena cava. Both of those go into the right atrium. Now, there are our usual cast of characters as far as the serous membranes, the parietal and visceral pericardium, but we also found that there's a third. It's not part of the serous membrane, but it is a very important sac around the heart called the fibrous pericardium. It's very protective and keeps it locked in its place. One of the more confusing things was understanding that if I am looking at membranes, I have a visceral and parietal pericardium, but that visceral pericardium has another name if I'm naming the walls of the heart. If I'm naming the walls of the heart from the inside out, it's endocardium, myocardium, epicardium. This epicardium is also the visceral peritoneum. It depends on what I'm naming. If I'm naming the walls of the heart, it is epicardium. If I'm naming the serous membranes, it's the visceral pericardium. All right. I know that's kind of weird. Now, the only type of muscle in the heart is called cardiac muscle. Right? Now, between the ventricles, we have the interventricular septum. Again, I'm not going to go through all of this, but chordae tendineae are what work the AV valves, atrioventricular valves. Off of the aorta, right on the outside portion of the aortic semilunar valve, we have the start of the left and right coronary arteries, which are bringing blood to the heart muscle itself, bringing oxygenated blood, I should say, to the heart muscle itself. And the coronary sinus is where that blood that has been used by the heart muscle gets drained back into the right atrium. Now, probably of more importance, that's all good, but probably of more importance to us on this is going to be really about just terms too. Systole versus diastole. Systole is contraction, diastole is relaxation. So when we're looking at a cardiac cycle, you need to understand that it is right at the moment that the ventricles and everything relaxes for that split second before it starts to contract again. Once it starts to contract, that's the start of the next cardiac cycle. Right? And so when we look at this, we tie it together with the fact that it has this, let me get back down here. It has these sounds that go in with it. Lub and dub, S1, S2, first sound, second sound. So the first sound is the sound of ventricle contraction. When the ventricles contract, you should know that means it's pushing blood out. I am not filling the ventricles by them contracting. Right? That if I asked you something about that, and it wasn't in the context of what we're talking about in here, if I said, "Can I borrow your water bottle?" If I said, "All right, I am going to squeeze this water bottle. I'm going to take the top off and I'm going to squeeze this water bottle shut," does that mean I'm filling it? No. That's just stupid to think any other way. But when we put it in this, it's like, I don't understand. Ventricles contract. That means it's filling. No, when the ventricle contracts, I'm pushing blood out of the heart. If I'm pushing blood out of the heart, it's going into two things: pulmonary trunk, aorta. That means those are the valves that are open. And if we're talking about sounds, that means the valves that are shutting are the ones we hear. If the semilunar valves are opening, that means the AV valves are shutting. Right? Again, it's a thought process that are simple steps along the way, but if you can overthink it, you can mess it up. But it's simple. Ventricles contract. That means I'm pushing blood out. As long as I know that the atrioventricular valves are the ones that are going to close when I'm pushing blood out, I know that's the sound. So S1, which is lub, is the sound of the AV valves closing. The second sound is when the ventricles start to relax. When the ventricles start to relax, that's when I'm filling the ventricles. That means that the AV valves, the bicuspid and tricuspid valves, open, and that means that the semilunar valves are the ones that shut. So the second sound, the dub, is the sound of the semilunar valves. This is the sound of systole, contraction. This is the sound of diastole, relaxation. Filling, ejecting. Anytime I hear something that is abnormal, when I'm listening for this, and by the way, listening to heart or listening to body sounds is called auscultation, of course. If I hear any abnormal sounds, that's called a murmur.
notice like any like abnormal sound?
Pretty much. And when you're listening to the specific areas, because that's the main sound you're hearing, it will definitely be a murmur. EKG and ECG, there are 3 waves to this. It's a terrible picture. That picture, this is not the picture. P wave, atrial depolarization. The atrium is pushing the last little bit of blood that's not going willingly into the ventricle, so it contracts, systole. QRS, this is ventricle contraction. Now, underneath this, there is atrial relaxation, atrial diastole, but it is totally swamped by the QRS wave from the ventricle systole. And then finally, the T wave, I'm getting all my letters mixed up, is going to be the ventricle relaxing, ventricle diastole. So that's the parts of the EKG, ECG. Now, there is, and I skipped it again, there is a pathway. You do need to know for sure the SA node is the pacemaker of the heart. The SA node is where the heart contraction starts. It's going to go from the SA node to the AV node through a little section called the AV bundle, which slows it down just a little bit. And then we go to the right and left bundle branches that come back around on what are called Purkinje fibers, which go into these little papillary muscles and cause the whole ventricle to contract and squeeze shut. Now, this kind of goes back to the idea of our body's monitoring stuff all around us. And so we're monitoring blood pressure, we're monitoring O2 levels when we looked at the respiratory system. But in the medulla oblongata, just like there was a respiratory center there with a ventral and dorsal control, there is a cardiac center. It is going to be looking for activity in our body, getting ready to start increasing contraction based on demand for oxygen. And what the things it's looking for are physical exercise. It can tell when we start moving. It can tell our body temperature is raised. If we get scared, this also triggers this to the muscle of the heart to start beating faster, and also concentration of ions, and this would also include oxygen as well. All right? So there are a lot of things that are monitoring it, and in our body we have dual control of this organ. We've got sympathetic versus parasympathetic. Parasympathetic is normal daily life, sympathetic is stress. Parasympathetic is going to be the vagus nerve, sympathetic is a bunch of other nerves. All right? In the heart, the parasympathetic is going to cause it to slow down. In the sympathetic, it causes it to speed up. So when you get scared, I think everybody knows your heart starts beating harder. All right. We have baroreceptors in our body, which are just basically modified mechanoreceptors, which are just designed to look for blood pressure. And so they are going to be in different areas. One is going to be, the two main ones are going to be the aortic arch and the carotid sinus. Aortic arch is where I can measure blood pressure into everything that's going out to my body. And in the carotid sinus is where I measure the blood pressure going into my brain. Now there are more than just it, but those are the main ones. And we're not going to get into this complicated reflex that goes through this. The blood vessels. Blood vessels are going to have 3 layers. like an ogre. And so it's going to have 3 layers. They're called tunics. Tunica externa, tunica media, tunica interna. In arteries, the tunica media is much thicker because it has a lot of smooth muscle. In veins, the tunica interna is going to have valves because there's not a lot of pressure in there, and the valves help things move in the direction desired. So arteries, again, carry blood away from the heart. As they start branching, they get smaller and become called arterioles. Arterioles are going to have this thing called a meta-arteriole, which crosses over to the venules, and off of the meta-arteriole we have capillaries. Capillaries are the only area in this that has a 1-cell layer thick wall. That is why it is permeable to plasma and interstitial fluid, because it doesn't have the tunics. As soon as the blood goes through the capillaries, it goes into small little venules on the venous system. These venules will start collecting and combining together to form larger vessels called veins. And again, arteries carry blood away from the heart, veins carry blood to the heart. So even if we look at this as the respiratory system in the pulmonary circuit, I've got arteries that are carrying blood away from the heart. It's going to go and branch into the lungs, into the small little capillaries. There they're going to have gas exchange, and in this type it's going to be between the blood and the air, and then it's going to go on venules out to the heart, which will collect together to go into veins, and then goes into the heart. It's still the same system. Now, the capillaries themselves, as they come off this meta-arteriole that goes from one side to the other, on the arterial side, and this we're just looking at the tissues, we're looking at the internal respiration. So on this arterial side, we're going to have plasma seep out, and it's going to be carrying oxygen. And it is the force of, I put on the wrong thing. It is the force of the blood pressure inside of it that is pushing this out. We call that filtration. So I start increasing the fluid in here in this interstitial fluid. As that happens, it's dropping off oxygen, and it's creating, as it's moving through here, it's creating a much more concentrated fluid, which is going to cause the interstitial fluid to be pulled back in, carrying its carbon dioxide by osmosis. These capillaries are going to be regulated in how they work depending on the tissue environment. If this tissue becomes more active, I need to get more blood to it. So it is controlled by what are called paracapillary sphincters, and they are only found on the arterial side. If this is just normal resting tissue, these will be closed down fairly tight. Still want blood to go through there, but I don't need a ton. If this is an area that starts becoming active, these will open up, more blood rushes in, I drop off more oxygen to the tissues that need it. Now, just to point this out, we didn't talk about this a lot when we first went through it. This is what most capillaries are, but there are 2 different types of modified ones. Fenestrated, these are the ones that are in the kidneys for sure. This is what we looked at. The glomerulus is a fenestrated capillary bed, which means I'm going to be able to have a lot more fluid move through there. And then in the liver, we have it to where these are not anymore just small holes, these are big holes. And that allows for, in the liver, to have a much bigger give and take of the blood that's in there. Again, not anything really major on that, just pointing out stuff that we kind of covered since we've gone through this. Now, veins are called the blood reservoir because at rest, most of our blood is in the veins. Up to 70% of all your blood is sitting in the veins as it's moving through. This is one of the reasons why I'm pretty sure in here we had, I can't remember, but I think there was a deep vein thrombosis section in here. No, it was only in one of the classes. But this is one of the problems, is because if I have this being my blood reservoir and I become sedentary, this is going to be ripe for blood. All blood has to do is sit still for a little bit, and then the clotting mechanisms will start. And so that's why you wear compression socks. And like if you're flying in a plane, they want you to get up and kind of walk around every now and then, because the pressure difference in there is going to cause our blood to kind of sit and pool for just a short period of time, and then it'll clot. Yeah.
This is a dumb question. Can you feel clots?
Depends on where they are. Yeah, you could probably feel where they are. I don't know if you'd feel the actual clot, but you'd feel the hardening of the arteries or the vein behind it and the pressure that's built up.
Would that be like painful?
Oh yeah. In general, it would be. All right, so blood pressure. Remember, you don't have to spell this, but it is called a sphygmomanometer. It's the blood pressure cuff. It is measured in millimeters of mercury. What you are measuring is atrial systolic pressure. Remember, it's the pressure from the atrium, or the arterial system, which is the pressure from the ventricles as they squeeze. And so ventricle systolic is when the ventricles contract. We're seeing the high spike in pressure in the atrial system. Arterial, I said. And then when it slows down, the diastolic pressure is the low part. That is when the ventricles are going through diastole part. So those are the only things I want you to really make sure you have in the back of your brain with that. Pulses are where you can feel the difference somewhere along the way. So we have stroke volume, which is how much blood leaves the heart for each contraction, and cardiac output, which is if I take that and multiply it by a minute. Remember that when we talk about blood volume, it is pretty common for people to just use this idea of 5 liters. It is the high end for females and the low end for males, but the more accurate description is it is a percentage of body weight. 8% of your body weight is going to be blood. And so when we're talking about blood pressure, there are things that we need to know about blood pressure, and not just at this point we're learning it just because we just want to make sure we pass the test, but understand this is what our body is trying to do, like we talked about with the renin-angiotensin system. That part of it is I need to control blood pressure. I need to raise blood pressure. So these are things that go over how our body would change blood pressure, factors that would change it. One is peripheral resistance. One of the big things in peripheral resistance is the control of the diameter of the arteries. So if I need to raise blood pressure, I will vasoconstrict. That's what that angiotensin 2 does. If I need to lower blood pressure, I vasodilate. I open it up. I lower the blood pressure. I'm controlling resistance by that. And that helps control my blood pressure. Viscosity. Viscosity is the thickness. Now, in general, this is a sign of dehydration, but there's other things that can go on with that. But the thickness of the blood is going to cause, if it's really thick, it's going to cause me to have to squeeze harder, to push harder, increasing blood pressure. If my blood viscosity is too low, my body is going to have low blood pressure because I don't have any resistance against it. Again, so this kind of is something our body is trying to monitor, but there's things that we can't change on some of it. We can control it, part of it, by cardiac output. Again, if all of a sudden my heart starts beating faster, I'm going to increase blood pressure because I'm putting more blood into the system. I can increase or decrease stroke volume to change it. Now, peripheral resistance, like we said, the changing the diameter is going to be a big thing that's going to be controlled again by the medulla oblongata. When we talk about the blood that's going back to the heart on the venous system, it is going to rely on skeletal muscle, and in the thoracic cavity, because it's not between muscle in there, it's relying on the change of pressure of my thoracic cavity in the idea of breathing. Right? All of these things are really kind of nice in the fact that if I need to get more of the blood out of the venous system to carry oxygen, which means I'm increasing my demand for oxygen, that generally means I'm active. If I'm active, I'm increasing my skeletal muscle contraction. If I'm active, I'm going to increase my breathing rate. Both of those kind of contribute to putting more blood in the arterial system. So it kind of is this little fail-safe to make sure we can have the appropriate amount of blood. So our oxygen. So that's why we have those veins, in order to keep things moving back towards the heart. Now, again, we've got the pulmonary circuit. Remember that when we talk about the pulmonary circuit, we start with the ventricle. Right? So the right ventricle, generally this is one of the harder things to kind of get because we're so used to grouping the atrium and ventricle together. This, we separate them. So the right ventricle is the pump, so it's the start of the pulmonary circuit. The left atrium is the last part of it. So if we look at the systemic circuit, we go from the left ventricle and then we end in the right atrium. Again, since the ventricle is the pump, when we're looking at the actual circuit, it's the driver, it's the engine, it's the start. All right. Can't forget the lymphatic system. I did have it, just didn't say. All right, so the lymphatic system. Again, this is kind of a cousin, it's a part of the circulatory system. So when we go back to that little capillary bed and we're pushing out the plasma, and then osmosis on the other end is pulling that back, that interstitial fluid back in, there's a percentage of that little merry-go-round that doesn't make it all the way through. So we have this buildup of pressure in the tissues. As that happens, we have these one-way capillaries called lymphatic capillaries. They have special spring-loaded cells that allow them to have this trap door to relieve the pressure that's increasing. And because it opens up a gap between the cells, larger molecules and things like bacteria and virus can get swept into the lymphatic system. At that point, the fluid is called lymph. So we have plasma in the circulatory system, interstitial fluid in between the cells, and then once it leaves and gets into the lymphatic system, it is then called lymph. And at that point, the lymph is going to be filtered through as it's bringing it back to the circulatory system. So it starts in the little capillary beds, it moves its way back, and it's going to go back into the venous system right before it goes into the heart. As it is being pushed through, and again, the lymphatic vessels are going to be very similar to veins, almost identical, and they have valves in it, same kind of mechanisms to get the lymph flowing as well, skeletal system and respiratory system. Now, as it is being pushed back to where it's going to come back into the circulatory system, it's going to go through little filtering stations called lymph nodes. In those lymph nodes, you're going to have little pockets that are going to be high with macrophages and T cells, memory T cells. Their job is to look for the things that are going through there, and if they have antigens that are not you, it will cause a reaction. It will cause an immune response, right? And so this is what we kind of refer to as the immune system. Now, there is one set of capillaries that is different, that is kind of unique in the fact that it is in the digestive system, and it doesn't say it here. I thought it did. They're called lacteals, and they're called lacteals because dietary lipids, the fatty acids as we break them down, the building blocks, are too big to get into a normal capillary bed. And so we rely on those spring-loaded cells from the lymphatic system to allow it to get in. So we have these things called lacteals that are in the digestive system to absorb lipids. And that's that. So. All right. So the only thing I want you to know about these vessels as they come in is that there are two ending ducts, thoracic duct and right lymphatic duct. The right lymphatic duct is everything above the diaphragm to the right, and the thoracic duct is everything else. So the thoracic duct is everything above the diaphragm to the left and everything below the diaphragm. Now again, this lymphatic fluid is going to be pretty much like tissue fluid. It's going to be exactly the same as it kind of gets pulled in. And the only difference between tissue fluid and lymphatic fluid versus plasma is the fact that plasma has those plasma proteins, specifically albumin, that's designed to stay in there. Now, if I have a problem where I'm not getting that excess tissue fluid that's building up to get into the lymphatic system, it'll cause an abnormal swelling that we call edema. Now, probably should just stick to just going through the review packet instead of trying to do it. So, in the lymphatic system, we have two types of tissues. We have lymphatic organs versus non-lymphatic organs, which are called MALT, mucus-associated lymphoid tissue. The difference is lymphatic organs have a capsule, these do not. These are mainly tonsils, that's what we would commonly think of. You and I, just normal people would think of tonsils. There are things like the appendix and different things like the prior patches along the digestive tract. But the three lymphatic organs are all of the lymph nodes, the thymus, and the spleen. And the spleen is the largest lymphatic lymph node, or the spleen is the largest lymphatic organ. Now, the big part of this is going to be getting into this part. Innate versus specific. Innate and non-specific versus adaptive and specific. So, our immune system is made up of these two lines of defense. One, you're born with. Right out of the bat, you have these built-in, which means innate, mechanisms. They are going to fight all pathogens the same. Anything that's trying to push you away from homeostasis, they have the same basic mechanism, which is going to work for most things. We also have a second line called the adaptive or specific defense. This is what we truly call our immune system. This is going to look for special markers on molecules, special molecules on things like bacteria or viruses that will cause a very specific reaction. It's called specific because these cells will not attack anything that they're not designed to attack. And it's called adaptive because you don't start off with it. Your body adapts to the things that are brought into you. Now, with this innate defense, The main one is this thing called a mechanical barrier, right? Your skin and mucous membranes. This is the first line of defense. And this is why when we talked about the digestive system and the respiratory system, we could say that things are still external until they cross over the membrane. And so what I eat is external all the way through me. Only the things that get absorbed are internal, right? Just like the air I breathe. And so we have this mechanical barrier that separates us from our surroundings. And this is truly the first line of defense. Nothing can affect you until it breaches this defense. I don't care what people say about different stuff, it can't affect you until it crosses this line. Does that make sense? And so that is why it's the first line of defense. Now, I don't care. I mean, I'm not going to ask you about everything else being called the second line of defense, but the first line is your skin. You do have chemicals that are going to help defend. I'm not worried about that. Natural killer cells are a type of a lymphocyte which doesn't become specialized. In general, lymphocytes are going to form either B or T cells, and they will be the part of the adaptive or specific immune system. But these cells, for whatever reason, drop out of school. They do not get specialized. They just want to go and fight. Phagocytosis, again, neutrophils and monocytes, which are also called macrophages, are the two phagocytic cells. I'm not worried about you knowing that they make up this mononuclear phagocytic system, but I do want you to know neutrophils and monocytes, which are also called macrophages, are the two phagocytic cells in your body. Fever is also a part of this innate defense. We're going to go through a cycle that's going to cause my temperature to raise. The two big things with this is, one, it is going to cause the viruses to not be able to replicate. The interferon is the name of the chemical. It's going to stop them from being able to make copies of themselves. The other thing that I just want you to know is the high temperature makes those neutrophils and monocytes / macrophages become more active or more hungry. Then we get into the adaptive immunity. Now, adaptive immunity is going to have molecules, look for molecules that enter our system called antigens. So we've got this first line of defense, and just underneath the first line of defense, I have all of these cells watching to see if anything breaks over. Once something comes over, then we're going to look at what's on the cell, and we can tell if it has an antigen that is not familiar with us, and we know that it means it is something that is trying to hurt us. So antigens generally mean a pathogen. Now, once that happens, we're going to have two defense mechanisms then activated. One's called cellular immune response, one's called humoral immune response. Cellular immune response is going to be T cells, humoral immune response are B cells. Now it all starts with this idea of something called a antigen presenting cell. It's going to see it, it's going to engulf it, it's going to present this antigen to different cells and ask, basically, do you recognize it? If it is something, first of all, if it's something that we know of, if it's something we've already come in contact with, we will have memory cells for that antigen. As soon as that happens, those memory cells, for this is T cells, these memory T cells will start making copies of themselves. At that point, one of the copies are called helper T cells. Helper T cells organize this whole response system. Not only just T cells, but B cells as well. Helper T cells are what are going to get everything going. Another type of cell that's produced when it's making clones is called cytotoxic T cell. This is the cell that's going to go out and actually try to find and bind with that antigen that's trying to invade us and attack us. And so cytotoxic T cells go out to fight. Helper T cells they're basically the generals trying to recruit and organize everything. Another thing that's going to happen is we will make more memory cells as well. So I'm going to start making different clones of myself, and there'll be 3 types. One type's a memory to stay back, one type's a cytotoxic to go fight, one is a helper cell to go and organize. If we're looking at B cells, once they get activated and start making clones, it basically is only going to make 2. I'm going to make other memory cells, and then I'm going to make what are called plasma cells. B cells' entire job is simply to make antibodies. It's going to start making these little protein gamma globulins and start putting them out in the millions into our bloodstream, which means they're eventually going to get into the interstitial fluid, intercellular fluid, all throughout our body. They are going to be looking for anywhere that there is one of these antigens, and they'll bind to it. Now, antibodies, there are 5 types of antibodies: IgG, IgA, IgM, IgD, IgE. IgG is the most common. Definitely want you to know that. And then IgM, because we're talking about the IgM antibodies are the anti-A and anti-B category of antibodies, so it'd be good to know that because that gets brought up a ton. All right. I'm not going to go over these terms. I'm thinking that you would be able to remember them if you looked at them. Agglutination's clumping, precipitation's falling out of solution, neutralization means it's no longer harmful. Chemotaxis is when we're calling something over. Opsonization means that I'm making it look tasty. And lysis just means breaking it down. So I guess I did go over, but anyway. In this idea that we just went over, we already had memory cells, so we looked at the idea of what would happen if we had an antigen come in that we were already familiar with. But if that wasn't the case, if it was its first time for us to notice it, we would call that a primary immune response. It takes longer because our body has to figure out what the heck is going on. How do we fight this? What's the best avenue of attack? What do we need to do? So this will end up taking a lot longer, and it's not as aggressive because, again, it doesn't know how to fight it right off the bat. But once this happens and I make memory cells, those memory cells provide me immunity for future contaminations, from future attack. At that point, we call it a secondary immune response. Secondary immune response, because I no longer have to study this antigen, is a very quick and very strong response. Now, secondary immune responses are anytime. So if you've had the flu before, anytime you get the flu after that, I don't care if it's 100 times, dear Lord, I hope it's not, but just in case it is, every one of those is considered a secondary immune response. It's only the first time we see it that is the primary. Right? But once we have those memory cells, then our immune system's ready for it to happen again. We have different classifications of that's it. We have different classifications of immunity. And so this goes to look at the difference between artificial and natural, and active and passive. Naturally means that it happens in nature. I don't need any scientist to make this happen. Artificial means it happens in a lab. Active means my body actually does something. There's some work to be done. Passive means there's not. These two are the same terms all the time we're talking about. Even when we're going in 210, talking about how things move across the cell membrane. You have active and passive transport. One is going to require energy, one doesn't. That's the exact same thing. In breathing, breathing in is an active, inspiration is an active process. Breathing out, expiration is a passive process. So those go back to the same things that we're always talking about. So it's really about natural, artificial. So if I have naturally acquired active immunity, that means that I become infected with a disease, my body fights it and creates its own memory cells. If I have an artificially acquired active immunity, that is basically the normal type of vaccine, where I have something that's put in my body that has been chemically altered so it's not as dangerous, and the idea is our body's going to attack it and make memory cells from it, so I'm still active even though it is coming from a syringe or whatever. Naturally acquired passive, the main thing with this is going to be breastfeeding, right? It is the way nature has it intended. That baby doesn't do anything. The mom's the one that produces the immunity. The baby just enjoys it. And then finally, artificially acquired passive means that we're going to have someone who cannot make their own antibodies, even if we put a attenuated, it's called, or a harmless version of the pathogen. Our body still is not in the position where I can create my own antibodies. So this actually just goes straight to the source and starts injecting someone else's antibodies into that system. So those are the different types. We can have it to where our body goes completely haywire and over-responds. We call that hypersensitivity. Now I want to just go down to, that's different than autoimmunity, where our body attacks itself. Hypersensitivity reactions are when our body is fighting something that should be a normal issue, but it just goes completely crazy. And so we have 4 types. Remember, only type 4 deals with T cells. These 3 are all about B cells and their antibodies that they're producing. Type 1, this is allergies. It is, again, this is way deeper than what I would ask, but I hope you remember allergies, IgE, eosinophils, all of those are associated with allergies. IgE and eosinophil, allergy, right? Type 2, this is what we looked at with blood transfusion, like we have if I am blood type A and I get B type blood, my anti-B antibodies will attack it. And then type 3, this is going to be rheumatoid arthritis, where our body doesn't use the antigens doesn't use the antibodies correctly, and we can't clean out where it's supposed to be. And then finally, like I said, type 4 are called delayed reactions. This is all about T cells. And it's called delayed reaction is because you have to wait to see if it happens. And if you've had a TB test, that's what they're looking for, is a type 4 delayed reaction. They inject you with a little bit, and it takes a little while. You come back a day or 2 later to see if there's a reaction. And so autoimmune, look, I'm not going to go through, I don't think, we're not going to have any of these tests on it or diseases on it, but autoimmune disorder is where our body just attacks ourself for whatever reason. Whether it is, one of the common things is if you had strep before, it will cause your body to kind of lose its mind and start attacking different parts. All right, trying. Digestive system. So the digestive system is this long tube that in the process is going to have different sections that will do different things The idea is I'm going to put material in this part of the tube, and as it goes through, my body's going to do special things to break it down. One's called mechanical digestion, that only happens in the mouth. The other is called chemical digestion, which happens at a couple different places along the way. But it's designed to break down food, break down this material we give it to the smallest possible part, the building blocks. That's why it's important to know carbohydrates are monosaccharides, proteins are amino acids, lipids are fatty acids, triglycerides and then fatty acids, and then nucleic acids are nucleotides. Those are their building blocks. We've got to break it down to the building blocks to absorb it. Now, we start with mechanical digestion that is going to be in the oral cavity. As that happens, we chew it and break it down. It's kind of funny. I did have a student in my 112 class this semester that sent me an email. It's like, because we talked about how you need to chew your food, and he's like, I can't, it's weird. I can't sit there. I keep thinking I got to chew my food more, and now I can't eat without thinking about it, and it's weird, you know. I'm like, I'm sorry. You should anyway. So this tube is called the alimentary canal. It is going to have 4 layers to it along the way. The mucosa is the inner lining, submucosa is just underneath that. Then you have the muscularis, which is the muscular portion that has 2 layers. In the stomach there is a 3rd layer. And then finally the outer layer called the serosa, which in the area below the diaphragm, the serosa is called the visceral peritoneum. Along the way, there's going to be these accessory organs. We start off with, again, now this is the alimentary canal, these different areas. So we chew, we swallow through the esophagus, it's stored in the stomach, and we put the battery acid that we call hydrochloric acid in there, start mixing it all together. We go into this, the largest part, which is the small intestines. Right off the bat, we have the combination of liver, gallbladder, and the pancreas, which dump enzymes into it. The small intestines is where most of the magic happens here. This is where most of the chemical breakdown to the building blocks happen and absorption happens. Then we get into the large intestines, which is going to reabsorb water. Again, water is kind of the key to everything. The stuff that is in the small intestines is going to be very watery, because water is what drives chemical reactions in order to break things down. All right, so if we talk about catabolism is done by hydrolysis, removing water. Or breaking down using water. And so that happens, and so we got to use the large intestines to take that water back. So as we look at how this works, there are 2 types of movements. Remember, we have a mixing movement called segmentation. This is mainly in the small intestines. Then we have the more popular or more commonly known propelling movement called peristalsis. This is moving things through this tube. There's a little wave-like motion of contraction that happens to propel it. Now, in this system, this is one of the few things that the parasympathetic increases activity and the sympathetic decreases activity. So it's almost like everything else is separate from the digestive system. Parasympathetic is going to inhibit most everything else, but it is stimulatory to the digestive system. The sympathetic nervous system is going to increase, stimulate everything else, but when it comes to the digestive system, it slows it down, shuts it off, actually. So mastication is the term used to talk about chewing, breaking things down. It's going to happen in the oral cavity. This mastication is mechanical digestion. Now, you should know that the palate is the roof of your mouth, the hard palate and the soft palate. I'm not worried about the idea of swallowing. There are salivary glands that open up into the oral cavity. This is going to start part of the chemical digestion. But the main thing is so that we can kind of combine all of this, whatever we're chewing, combine it together to start the process of breaking down. And again, we probably should chew a lot more than we usually do. Now, from the oral cavity, we open into the pharynx. Again, we've looked at this many times. The pharynx has three parts. The part behind the nasal cavity is the nasopharynx. The part behind the oral cavity is the oropharynx. The part behind the larynx, which is this thing, is the laryngenopharynx. At the laryngenopharynx, there is a dividing line. Things that are going to the stomach are going to go posterior through a tube called the esophagus. The esophagus has no digestive properties. It is simply a tube to get that which is called bolus that I've chewed down to my stomach. All right. It is going to use peristaltic waves to get it down there. There is our first sphincter that we look at between the esophagus and the stomach, called the lower esophageal sphincter. That's what I'm going to call it. Now, once it enters into the stomach has got another sphincter on the other end called the pyloric sphincter. So we make this little cavity, we make this little pouch to store the food that we've eaten in. The stomach is going to then mix it with two particular things. We have pepsinogen, which is released, is inactive. It's released as pepsinogen from chief cells, and it becomes active to form pepsin. This is designed to help start breaking down and unraveling the proteins. We're also going to have hydrochloric acid, which is going to come from parietal cells. This hydrochloric acid is what converts pepsinogen to pepsin. I'm not worried about gastric lipase right now. I'm hoping you know mucus on that. Now, I'm going to come back to that in a second. So the control of the stomach's weird. So we can have it to where it's controlled before anything gets into it. This is called the cephalic phase. This is the idea of looking at something, smelling something, or even hearing something will cause your stomach to start rumbling, to get ready for what's going to come down to it. The gastric phase is what's going to happen when we have things in the stomach. There are certain things that help positive feedback almost, where it helps make sure I'm doing things to help what's in the stomach. Once I've started letting things get into the small intestines, then we have intestinal control. The intestines will kind of send a signal back to the stomach and say, slow down a little bit, let me work on what you just gave me, and then we'll go from there. So when we talk about this, we have control. I should go back up to this. In the stomach, during this gastric phase, we will have something called gastrin that is going to be released in the cephalic and the gastric phase that cause the stomach to become active, to create more gastric juice, to create more fluid in there and get ready for what's going to be coming in. This third one down here, this cholecystokinin, is really from the intestinal phase. It's designed to slow the stomach down. It's designed to cause it to kind of halt a little bit. Now, once we get this into the small intestines, right? So as the stomach's churning, it's going to open up. Now again, when I'm chewing food, it's called bolus. Once this gets in the stomach and starts mixing with the gastric juice, it's called chyme. Once this chyme gets kind of mixed to where our stomach thinks it's good, it will start slowly opening that pyloric sphincter and letting small amounts get into the small intestines. So in the small intestines, we have three sections: the duodenum, the jejunum, and the ileum. DJI. The duodenum, the jejunum, and the ileum are the three sections of the small intestines. The duodenum, right at the start, I have the liver, which is producing bile that's stored in the gallbladder joining, and then I have the pancreas right at the start. Now these are going to secrete different things that are going to be put into the chyme so that when the segmentation happens, it starts breaking down all the chemicals. So the pancreas has got both an endocrine and exocrine portion. The endocrine is insulin and glucagon, like we looked at the end here. Exocrine is going to be this stuff called pancreatic juice. In the pancreatic juice, we're going to have enzymes that break down all 4 of the organic molecules. We will have things, amylase works on amylose, which is a type of carbohydrate. Lipase works on lipids. Trypsin, chymotrypsin, and carboxypeptidase all work on proteins. And I said generally it's going to be things that work on protein are going to be have one of 2 things. Either they're not going to end in ASE, or even if they do, they will have something about peptide in the name. All right? So it's either not going to end in ASE. If it doesn't end in ASE, that is a protein enzyme. And if it has peptide in it anywhere, it doesn't matter if it has ASE or not, that means it's working on proteins because it's breaking down the peptide bonds. And then finally, nucleases, which are breaking down nucleic acids. Now, the other part that it is doing, the other section that it is going to be creating stuff, is going to be creating this bicarbonate ion. Again, bicarbonate ions are our buffer system. The stomach has a very low pH, or said another way, a very high acidity. Once it enters into the duodenum, I need to get that pH back up to a normal, handleable section. So it is bicarbonate that's released to help raise that pH to where it needs to be. Now, the way our body controls the pancreas is 2 things. One of them goes back to our buddy cholecystokinin. Cholecystokinin is released, causes the stomach to slow down, and it also causes the pancreas to contract and release the digestive enzymes. Secretin is what is released by the small intestines to help release bicarbonate. Secretin is the duodenum going, ow, acid, and it sends a signal to cause it to release bicarbonate to help get it back to where it needs to be, the pH. The other thing that the cholecystokinin does is it is also going to cause the gallbladder to contract and squeeze out what it is storing, and it is storing bile. Now, when we talk about this, bile is a huge thing. Bile is needed to break down or emulsify lipids. Again, in our body, it's mainly water, particularly in our small intestines. If we have fats, lipids, oil, whatever you want to call it, in that area, it will not break down normally. And so we need to have some sort of chemical that will break down the fat into small droplets, which we refer as science called emulsification. And so that's what bile does. Now again, I am not going to worry about micelles and all the different things. I do hope you understand, again, we still have a section of fat-soluble vitamins, vitamins A, D, E, and K. You also have water-soluble vitamins, which are B and C. I'm hoping you still remember that. So anything that's fat-soluble has to have bile in order for it to be properly absorbed. Now, the small intestines, again, is where most of the absorption happens. It is the longest part. Again, it's got 3 parts to it: duodenum, jejunum, and ileum. I do want you to know that there are villi. Now, I'm hoping you understand the idea of these Plycia circularis, which have villi that stick off of it, and the villi have cells on them that have microvilli on it. It is increasing surface area over and over. The main thing I want you to know are these villi, and the villi have the capillary bed along with this lymphatic capillary called the lacteal that are designed to absorb all the stuff that our body is breaking down. All of that outside of the lipids that are going into the lacteal. But this capillary bed that's absorbing all the amino acids, all the glucose or the monosaccharides and the different nucleic acids, all of this, or nucleotides, all of this is going to go to the liver, and then the liver is going to remodify everything, right? So that's where we talked about those two blood flows to the liver. Again, I am not planning on asking any of this, but I want you to make sure you understand, because it is something that I'm hoping you get. I've got oxygenated blood coming from the hepatic artery, but I have deoxygenated blood coming from the intestinal tract that has a bunch of nutrients, all the things we talked about that are being absorbed. This is the hepatic portal vein. Those merge. The hepatocytes, which are the liver cells, are going to modify and make sure all the things we absorbed are safe for us, convert all monosaccharides to glucose, and in the process, they'll start pulling out red blood cells that are marked for recycling and breaking it down. In the process of doing that, I make bile, right? And then the bile, again, is used to help break down lipids. All right. Now, the large intestines, at the end of the large intestines, or between the small intestines and the large intestines, I should say, is going to be this sphincter called the ileocecal sphincter. That's the dividing line between the large and small intestines. From that point, the large intestines is really about removing water. At this point, the stuff that's inside is called feces. It's bolus in the mouth. It is chyme once it enters into the stomach, and it is chyme until it gets into the large intestines, and at that point, it's called feces. All right. Now, again, I'm trying to cover things, and sorry. I hope you understand that about the internal and external anal sphincter, and that, again, basically the large intestines is about removing the water. Once we get into the respiratory system, now we're looking at trying to get oxygen to unlock the power of the materials that we just digested. So in the respiratory system, it is all about gas exchange. Respiration means gas exchange. And I have an external respiration, which happens in the lungs. External respiration is when the carbon dioxide in the blood goes into the air and the oxygen in the air goes into the blood. Then it goes back to the heart, goes out to the body, and there we have internal respiration. Internal respiration is where the oxygen that's being held on by the blood is released, carbon dioxide gets pulled back in. And then we have cellular respiration, which uses the oxygen to break down the glucose efficiently to produce ATP. And remember, in the production of ATP, I am going to decrease oxygen levels because I'm using oxygen. I'm going to increase carbon dioxide because that's a byproduct. I am going to increase the acidity of the tissue and the heat in the tissue. Those are all signs of cellular respiration. And they're all signs, are all symptoms that will cause a much greater release of oxygen. So there's all these little checks and balances that go on. In the anatomy of this, above the larynx is called the upper respiratory system. Below the larynx is called the lower respiratory system. I'm going to get through this. I'll move through as much as I can. The pharynx, same thing that we looked at. Now, in this, we have an area, the laryngenopharynx, we get into the larynx, all right? So this now, we're in the digestive system, we're on the posterior side with the esophagus. This area called the larynx is the start of the bronchial tree. This is what's gonna hold your voice box, and it also is on the external, is gonna have two types of cartilage. Thyroid cartilage is the largest one. In males, again, it's more pronounced. That's why they call it an Adam's apple. The cricoid is the one that's a little below that. But underneath that, I start a tube called the trachea, which is commonly called the windpipe. Now, inside the larynx, though, we have this little area, like I said, the voice box, and there's going to be three structures here. An opening called the glottis, which is covered and protected by an elastic cartilage flap called the epiglottis. And on either side of the glottis, I have tissue called true vocal cords because they make noise, and then false vocal cords, which are a little bit farther to the side and above it, which protect the true vocal cords. They don't make noise, so they're called false vocal cords. Again, from there, the trachea, which is the windpipe, goes down, and it will start dividing into what, again, we call the bronchial tree. You have a right and left lung. The left lung is smaller because of the heart. It only has 2 lobes. The right lung has 3. As the bronchial tree enters, we have a primary bronchus that goes into the lung. So we have a right and left primary bronchi. From there, it will divide and go into the lobes. They can either be called secondary bronchi or lobar bronchi. Once it enters the lobe, it will divide into the segments that are inside the lobe, and it is called the tertiary or segmental bronchi at that point. As this bronchial tree gets smaller in diameter, there's going to be a decrease in the cartilage that's in there until it's gone. The cells that line it will go from columnar, they start off as pseudostratified columnar epithelium, to cuboidal, to finally squamous. The only thing that doesn't appear to decrease is smooth muscle. As it gets smaller, the ratio, the idea of how much smooth muscle is in it, stays relatively the same, if not increase a little bit. And this is to make sure that we can guide the air to where it needs to go. Now, at the end of this are these little hollow pockets called alveoli. Alveoli are the site of gas exchange. Now, with the alveoli, I'm going to get down a little farther here. I'll come back up to this. With the alveoli, we're going to have this thing called a respiratory membrane. The respiratory membrane is going to be made up of the simple squamous cells of the alveoli and its basement membrane, and then the simple squamous cells of the capillary and its basement membrane. So there are 4 layers to it. This is where external respiration happens. Now, this is the only place that external respiration happens in this system. So, when we look at it, we have what's called anatomic dead space, which is all the bronchial tree leading up to this. Anatomic dead space is normal. Everybody has it. However, if we have areas of the respiratory membrane or alveoli that aren't functioning, that is alveolar dead space. That is abnormal. That is pathology. If I want to know the system and how well it's working, I can add those 2 up, and that's called physiologic dead space. That's the normal plus the abnormal. Right? Now, in this area, there is going to be 2, in these alveoli, there's going to be 2 types of cells. Type 1 cells are just simple squamous epithelium. The type 2 cells produce this thing called surfactant. The only thing you need to know about surfactant is it reduces surface tension. I'm not going to go over what surface tension is, but I hope that you remember a little bit about what surface tension is. Now, when we talk about inspiration and expiration and breathing, it is the act of changing the pressure in our thoracic cavity by changing the size of it. That's why we look at this thing called Boyle's Law. Boyle's Law, which really doesn't talk about breathing, all Boyle's Law is a very general idea that anytime I decrease pressure or decrease volume, I'm going to increase pressure, or anytime I increase volume, I decrease pressure. It simply says that those 2 values are antagonistic, which means that if I increase one, the other one decreases, or vice versa. All Boyle's Law is. So what it's saying is, in order to change the pressure inside my thoracic cavity, which is what's going to move air in and out, I change the volume. And I do that with mainly the diaphragm. So the diaphragm is the main muscle of normal breathing, but I do want you to know, if I need to take a breath in that's bigger than normal, I'm going to use external intercostal muscles because I'm trying to get my ribs to move outward, and I can only pull, I can't push, so I have to be on the outside to pull it. And if I need to breathe out more forcefully, like sneezing or coughing, I am going to use my internal intercostal muscles to squeeze my thoracic cavity to push air out. Now, we look at the health of our lungs by measuring certain abilities for us to move air in and out. Now, this is called spirometry. The only thing I definitely want you to know is tidal volume. Tidal volume is normal breathing, and it is 500 milliliters. You don't have to know any other volumes for sure, but I hope that you have an idea of what these are, especially on this side. Tidal volume is normal breathing. I have inspiratory reserve, which is area I can breathe into, especially if I'm going to yawn. And then expiratory reserve if I need to blow more air out. This is air reserve for use. And then finally, I have this bottom value, which is the residual volume, which is air that's trapped in the tubes. Can't use it. Now, as we are going through this, remember that we have 3 areas of control. The pons, which has the pontine group, which is above. Remember, it is above and it sets the upper limit. It's basically over the top of the medulla oblongata, so it's looking down and it's saying you can't breathe any more in. It sets the upper limit. Ventral group controls tidal volume. Dorsal group is monitoring everything going on in your body and adjusting this to what it needs to be. So if I become more active, the dorsal group notices it and changes and talks to the ventral group and changes normal breathing. Now, I'm going to monitor my breathing with chemoreceptors in 2 areas. Central chemoreceptors are going to be in the central nervous system, peripheral in the peripheral nervous system. Central chemoreceptors, because I am just worried about the pH inside the central nervous system, I am worried about 2 things. This is monitoring hydrogen ion levels, which is directly pH, and it is measuring carbon dioxide levels, which is the future pH. Carbon dioxide acts as an acid in our body. So central chemoreceptors are looking for that. The peripheral chemoreceptors are actually going straight to the O2. These are monitoring the same 2 basic areas we looked at when we're talking about blood pressure. Carotid sinus, aortic arch. Ooh. Now, remember that in our body, and the only 2 gases we're really concerned about here are oxygen, carbon dioxide for us in here, that oxygen is almost entirely carried on hemoglobin, bound to hemoglobin. 98% minimum is bound to hemoglobin. So the main way oxygen gets transported in our body is bound to hemoglobin. This is very different from carbon dioxide. Now, the things that will cause hemoglobin to release oxygen are all the things that are involved in an increased metabolism. If I start becoming active, I'm going to use the oxygen that's around, so I'm going to decrease oxygen. I'm going to start producing carbon dioxide because that's what happens when I break down glucose, so I'm going to increase carbon dioxide. I'm going to increase heat, and I'm going to create an acidic environment because that's part of the process. As soon as my blood feels this, then hemoglobin is going to start wanting to let go more than just 1 oxygen. Because under normal circumstances, again, hemoglobin can only hold 4 oxygen molecules. Under normal circumstances, if all of this is normal, it's just going to release 1. Once this starts changing, it might release 2 or sometimes 3. Right? So that's what's controlling it. And that's why in our venous system, blood is considered 75% saturated because of the 4 possible areas that have oxygen, generally 1 is going to be released. Now, carbon dioxide, on the other hand, isn't as straightforward. Some of it dissolves in the plasma. CO2 dissolves in fluid a lot easier than oxygen. That's why we have carbonated beverages. Some of it binds to hemoglobin. Basically, the more I create, the more I have carbon dioxide, the more it wants to kick out oxygen. So again, it's this little fail-safe. But the majority of it, 70%, is going to be turned into what we call bicarbonate. Again, this is done so that as I'm carrying this waste product, I'm using it until I get to the dump, which is the lungs. So it's going to go through this process, going from carbon dioxide, binding to water, carbonic acid, break it down to bicarbonate and hydrogen ions. And so once it gets back to the lungs, that environment of the lungs, which is much different than inside the rest of our body, it causes this equation to reverse itself, and therefore we breathe out carbon dioxide and water. All right. Just closing in. The urinary system. So with the urinary system, now we're going to filter this blood. So we've used it to pull in the building blocks from the digestive system. We have used it to carry oxygen to the cells, also to use for energy. Now we're going to clean this fluid that we call blood in the urinary system. The kidneys are the workhorse. Inside the kidneys, we are going to have, remember that the kidneys also are retroperitoneal, which means they're behind the peritoneum. And the kidneys are going to have an outer layer called a cortex, which is where you're going to find the nephrons, and an inner layer called the medulla. Renal cortex, renal medulla. Inside the renal medulla is where you're going to find the renal pyramids and these little things in between them called renal columns. Now, because we're skipping through kind of to get through some of this, we have 2 types of nephrons. And again, remember this outer layer is called the cortex. So I'll have a group of nephrons that are going to be relatively close to this dividing line between the cortex and the medulla, renal cortex and renal medulla. Because of that, they're called juxtaglomerular or juxtamedular nephrons. They are going to have a very long nephron loop, and that nephron loop is going to have a very organized peritubercapillary bed called vasa recta. This is where we get the majority of this countercurrent multiplier. Now, there's only a small percentage that are here. Now, this is a small percentage of the renal cortex. The majority of the renal cortex is above this, and that's where we're going to find the cortical nephrons. Cortical nephrons have a relatively short nephron loop. They will still dip into the renal pyramid, but they are not going to go way deep. They still have peritubular capillary bed around it, but it's not as organized as those long ones. So when we're looking at the nephron, remember that what we're looking at is this filtering system. I have a capillary bed called the glomerulus in here. The blood bringing, the vessel bringing the blood to it is called the afferent arteriole. The blood taking, or the vessel taking it away is called the efferent arteriole. As it comes in through here, we are going to create filtrate. The filtrate is created mainly by the pressure of the blood that's inside the capillary, inside the glomerulus. That's going to be the main force, but we're going to have 2 that resist. One is going to be the fluid that's already in this space, and the other is going to be the fact that as I am losing this water called plasma, I'm going to create a much thicker, much more osmotically magnetic type fluid, and so that's going to resist it. Those three forces work together. Again, the main one is going to be what we call glomerular hydrostatic pressure, pushing the fluid out. That's going to create a fluid called filtrate. It then goes through this tube called the nephron loop, or the nephron tubule. I'm going to have the proximal convoluted tubule, which does 70% of all of the give and take. So this efferent arteriole that comes out, it's going to form a paratubular capillary bed around the whole thing. In that, I'm going to have, if I have the tube here, and I don't have a colored thing, but this is the blood here, and we'll just draw it going the same way. Things that are in my blood that didn't get filtered out that I want to get rid of, go from the blood into the tube. This is called tubular secretion. Things that are in the filtrate that I don't want to get rid of are pulled back into the blood by tubular reabsorption. The majority of all of that happens in the proximal convoluted tubule, but it doesn't change. It's going to do the same thing all the time. Then we get into this nephron loop. Remember that in the nephron loop, I have a descending and an ascending limb. The descending limb is permeable to water, but not ions. The ascending limb is permeable to ions, particularly sodium, but not water. All right? That's an important part to remember. Descending limb lets water out, the ascending limb will not. The descending limb will not let ions out, but the ascending limb will. We go through, as the ascending limb gets back into the renal cortex before it goes all crazy in the distal convoluted tubule, it's going to pass right by the afferent arteriole. We call that the juxtaglomerular apparatus. In that, I have two types of cells. Macula densa cells are the ones that are on the tubule. They're monitoring sodium levels. If I see that sodium is too high, it knows that the fluid is going too fast. If it's going too fast, it's going to vasoconstrict the afferent arteriole. That lowers the glomerular blood pressure or hydrostatic pressure. That lowers filtration rate. Basically slows down the creation. If I see that there's not enough sodium, I know it's going too slow. If that's the case, the macula densa cells talk to their buddy, the juxtaglomerular cells, and they vasoconstrict the efferent arteriole. That builds pressure and speeds up the creation. From that point, we get into the distal convoluted tubule. Now, the distal convoluted tubule, this is all about homeostasis. This is where our body can regulate things like hydration levels. So aldosterone and antidiuretic hormone are going to cause the reabsorption of water through different mechanisms. Or if I have the atrial nitritic peptide that the heart's sending out saying I have too much blood volume, it's going to affect here, causing us not to reabsorb water. Make sure that we pee a lot, get it all out of us. Now, the distal convoluted tubule will attach to a collecting duct. The collecting duct will have multiple nephrons attached to it, and it's still called filtrate at this point. Once the collecting duct gets to a point where it drops off its its fluid onto one of these things called a calyx, now we're talking about urine. So each one of these renal pyramids has a minor calyx at the point of it. If the minor calyxes come together, this is called a major calyx, but whatever the case may be, the end result is a big hollow room called the renal pelvis. The renal pelvis then turns into the ureter as it exits. The ureter takes the fluid, which is now called urine, to this thing called the urinary bladder. The urinary bladder is going to store the urine. It has a lining made up of transitional epithelium. Transitional epithelium changes shape. Real quick, Inside the floor of the urinary bladder is a little triangular shaped thing called the trigon, which doesn't have transitional epithelium. And it's because where the two ureters come in and the one urethra exits don't need to have a transitional epithelium around it. Not worried about the 4 layers, but you should know that the muscular layer of the urinary bladder is called the destruser muscle. Now, the urethra is going to be different compared to males and females. In females, it is a dedicated urinary tube, but it is short. In males, it's longer, and it has both urinary and reproductive values to it. And then the word micturition simply means going pee-pee. All right. Last 2. Endocrine. Where is my endocrine? So the endocrine system, going back to what we talked about with this, the endocrine system, just like we had for the test, is mainly going to be about hormones, glands. Hormones and glands. It will have a little bit about steroid, lipid-soluble hormones versus non-steroidal, water-soluble hormones. Those 2 areas are the big keys. Those would be the same things that I try to say to hone in on. Again, when we're taking this test, where I can ask more questions, it's a little different. In here, again, out of the 10 to 12 questions that might come from this chapter, I would say 7 to 8 of them will be from those 2 things. The majority of them will be basically knowing what glands secrete what hormone. Okay. You do need to know that endocrine system is made up of glands that don't have ducts. That's the whole point of they're an endocrine gland. What they secrete is called a hormone. Hormones are chemicals that are meant to go make a change somewhere. The endocrine system works hand in hand with the nervous system. What the nervous system doesn't control directly with nerves, it will control indirectly with hormones. And that hormones are specific, and what they act on are called target cells. And so something like human growth hormone or growth hormone, every cell is a target cell for it. But certain hormones are very specific. Thyroid stimulating hormone. The only thing that's going to be a target cell for that are thyroid cells, right? But hormones are what the endocrine system uses. That's basically it. So I wanted to get through that to get to the final part, which is looking at this part of the reproductive system that we looked at in the lab. All right, so with this, like I said, there's only a few things that I'm worried about you knowing on this because we didn't really have time to get into it. So with males, you need to know testicles, the testes, are the male sex gland as well as their endocrine gland. They produce a hormone called testosterone, but the sex cells are called sperm. Remember that sperm are reproductive cells, which means they have half the genetic material. Therefore, they are called haploid cells. Diploid cells are the ones that have full genetic material. In order to create haploid cells, they go through a process called meiosis. Meiosis is mitosis, basically done twice with a few little changes. The thing I want you to know about it is in males, meiosis is referred to as spermatogenesis. In females, meiosis is referred to as oogenesis. All right? So the testicles are going to make sperm in this, and they're going to be carried through this vas recta, that's in the nephron. The vas deferens or ductus deferens. The big thing I want you to know is for this, Only males have prostate glands. Prostate glands are kind of the merge center between the reproductive and urinary system for males. I want you to know that there's two types of tissue that are going to be in the penis. You have corpus cavernosum and corpus spongiosum. Corpus cavernosum is the erectile tissue. Corpus spongiosum surrounds the urethra, and once we get to the distal end, the corpus spongiosum is what forms the glans penis. Now for females, we have the ovary. The ovary is the sex gland for females as well as their endocrine gland. From the endocrine system, it produces estrogen and progesterone. It is going to also create oocytes. Oocytes are commonly called eggs. They are going to be caught by this thing called fimbriae and brought into the uterus by way of the uterine or fallopian tube. In the uterus, the inner lining is called the endometrium. Endometrium is the one that I want you to know about. The endometrium is going to be with the oocyte is fertilized. This is where it's going to be planted basically and grow into this little baby. It has an opening called the cervix that opens into this canal called the vagina. And the cervix, again, is where when the baby is ready to be born and it kind of twists, the head gets here, and as the head starts stretching the cervix, there is this positive feedback mechanism of this hormone called oxytocin, which comes from the posterior pituitary, and remember, that means it's made in the hypothalamus. This is going to cause the uterus to contract, which pushes the baby forward, causing it to stretch more, which causes more hormone to be released to cause more contraction. That's why you have contraction waves as it goes through. And that is it. Sorry, probably spent too much time on blood. I gotta stop that, sorry.